congratulations to those who got the Bethel Summer Scholarships. And it's a, it's a privilege, right? That you would be able to get that type of support to do research. So, in any event, Lori, Dr. Dierhoff there, she and Dr. Bean, you've been introduced to, yeah, they created this weekly seminar for the summer, and it's very valuable for you, especially the early trainees, to learn just some really basic skill sets in order to do research, right? To do effective research in a lab. So, in any event, I'm Professor Nam Vo. I'm a co-executive, what do you call it, my title, scientific director of this Bethel Musculoskeletal Research Center. And what I'm gonna do today, so what I'm gonna talk to you about today is how to do basic research, how to do laboratory experiments, okay? And how to keep a proper lab notebook, okay? So, there are different basic skills that you need to acquire in order to do effective research. And these are some of the skill sets that you need to acquire. One is how to read scientific papers, and Dr. Alexander last week coached you through that, right?
Skip.
What?
We skipped it.
Yeah. It takes a lot of time and practice to do all of these right. That's why a PhD takes about 5 to 7 years. And how to listen to a research talk, right? And how to write. That means writing, once you get enough data, you can write and publish. And the act of writing a scientific research paper or writing a research proposal, that you need to learn as well. How to write, and then how to present, right, your research data. So all of this takes time and proper practice to do it right and to do it effectively. So all of this you need to acquire in order to think, right, to communicate and to solve problems effectively. And a sort of a common trait of being a scientist is that they all seem to be addicted to finding things out. They're curious. Curiosity is what drives science, right? And that's our sort of innate desire to understand things around us that make us want to become scientists. Okay, so Dr. Peter Alexander show you how to do that, how to read scientific papers. Okay, then I'm going to talk about doing laboratory experiments, and Dr. Bridget Deasy will teach you how to present your result. Right? At the end, you're going to do a poster presentation, okay? And she's going to teach you how to do that. So there are two general types of presentation. One is a podium, meaning you stand up, right, and talk. Another is you have to print out on a large sheet of paper, a poster, your research data, and you present it that way. That's like somewhat similar, but there's slight differences between those two modes of presentation. Okay, so the question is, what is science? Okay, to a lay person out there, it might sound very esoteric and scary at times, but science, it boils down to, is a way to finding things out. It's just a technique. That's what it is. Okay. And so, for example, some of the questions or problems that we could use the so-called scientific method, right, to solve problems. How does a person get sick from eating food at, say, a restaurant, or why is my car not running today, right? Those are scientific questions that you can ask and solve. There are other non-scientific fields that has to do with emotions and some other domains of knowledge that cannot be tested, okay, or tested in a logical manner. So, for example, if you say, okay, this painting is beautiful or killing animals is sinful, those are not really scientific. It's hard to test those, okay? But what I'm going to show you or coach you through today is the scientific method, okay? In order to do science, you have to apply, in general, this method, this methodology to solve a problem or to answer a question, okay? Okay, so what is the scientific method? Okay, these are the basic steps. First, you have to start with a question. Right? What's the problem that you want to understand or you want to solve? So that's the first thing. Okay? So when you read, say, for example, a paper, a scientific research paper, in the introduction, you read through that, and if you cannot find or understand what the problem or what the question the authors try to address, then either you don't understand what you're reading, or the authors did a bad job of writing the paper. So you have to understand what the question is, right? And in general, the question is derived from some kind of observation, right? In general, okay? And then this is the third step is, from the question, then you just guess an answer to the best of your knowledge, to what you know, okay? And that's what's called a hypothesis. Hypothesis, just an educated guess, okay? And then you don't stop there. The next step is you have to demonstrate that your hypothesis or your answer is correct or not, okay? If you study philosophy, you can stop there. You make, or religion, you just make a statement, make a guess to whatever the question at hand, and you say, okay, that's good enough. You don't have to prove anything. But for it to be scientific, this is an important step. You have to provide some kind of evidence, okay, to show that your guess or your answer to the question at hand is correct or not correct. And that, it's a sort of a human revolution, like a big, step forward for humanity about 500 years ago. I forgot, some guys came up with that idea, which is extremely important, okay? So you have to do some kind of experiment to test your answer is correct, okay? And then, of course, you find that you find the result, and then you compare your result back to your answer, right? Your hypothesis, right? Do your findings support the hypothesis or not? If it doesn't, then you have to come up with another answer, okay? So there you go. Those are the basic, steps of the scientific method. If you look at that and try to memorize these, and I'm going to share these slides with you guys afterward, okay? All right, so get your answer, get your result. You have to compare it back to your hypothesis or answer, and then see if it supports your hypothesis or not. If it doesn't, you have to come up with another answer with a different hypothesis, okay? And this is very important, okay? If your experimental findings do not support your hypothesis, then the hypothesis, right, that hypothesis is wrong, okay? And it doesn't matter. This is the beauty about science. It doesn't matter if you're an important figure. If your hypothesis, is wrong, then it's wrong. okay? It doesn't matter if you're like some big shot. That's what Mother Nature tells you, right? You do the experiment, carefully, and it spits out some answers, and those findings do not support the hypothesis, then your idea is wrong, your answer is wrong. Then you have to come up with the next hypotheses. Okay, and you might, have this "so what" in your head answer. And this scientific method, right, it's what drives technology, right, drives medicine. Basically, if you look all around you, all the benefits that we experience now is due to this set of investigation, the scientific method. Okay, the food that you eat, we have plenty of food now to eat. If you go back just 50 or 100 years ago, we didn't have the technology to grow this much food. Where does that come from? It comes from the from science, from applying scientific method, right? You all have computers and cell phones now. You think those just like pop out, from thin air? No, that's like years and years of accumulated scientific knowledge, okay? So that's what the scientific methods allows you to do, to understand the world around you, right? And to generate fundamental knowledge and create a lot of wonderful product, okay? Very powerful product. Okay, so basically the scientific method is one of the most, if not the most important, okay, human discovery. It's the golden key that allow us to understand nature, phenomena around us. Okay. In fact, I put this thing on here. Humans evolved, around 5 million years ago. It's only about 500 years ago that's when the scientific method began. Okay, and that's how we have so much accelerated advancements in terms of everything, technology, right? Social, development, all the technologies, all the advancement that we enjoy now is due to this scientific method. So I'm going to give you an example of what a scientific method looks like, okay? And this is for a general person's example. Say, how many of you like fishing? There, only the boys. Okay, say you go on a charter fishing trip, okay? And then you made an observation. You couldn't find your wallet. Say, okay, my wallet's missing. That's an observation, right? And say, the question is, where's my wallet? Right? That's an important question, because if you were to lose, I don't know, a dime or a dollar, then you might not, who cares? But a wallet, that's important. So the question is, where's my wallet? So the hypothesis, right, now you have to come up with an answer. You say, maybe someone on the boat stole it, okay? Or I left it at home, right? Or I dropped it overboard. Right? All of these are reasonable guesses, right? Or the alien took it. Okay. Now, hypotheses make predictions. Okay? So the prediction is, if somebody on the boat stole it, then if I search the people on the boat, you would find it. Right? That's a prediction. And then if I left it at home, I would go home and would find it at home. And then if I drop it overboard, then if I search the ocean, it might take me 100 years or 1,000 years, then I might find it. Okay. If the alien took it, then I could find it if I find the aliens and search that. Now, what sets science apart is that The scientific hypotheses makes testable predictions. It's not just any predictions. It has to be testable, okay? If you cannot do experiments to test your prediction, then it's not scientific, period, okay? So, this is not testable. If you don't know, what an alien looks like or if they really exist. So that's not testable, so that's not scientific, okay? Drop it overboard. This is testable, but it's very hard to do. So sometimes we do science with the presence or the current technology that we have that allow us to test, to do the experiments. If it's too difficult, then it's nearly sort of pseudo-scientific because it's just hard to test. These two you can test easily, okay? All right, so let's go through. The question is, where's my wallet? My wallet is missing. That's based on that observation. And then the hypothesis is that I left my wallet at home. So the prediction is, if I go home, I search for my wallet, it should be there, right? Okay, so that's what you did. You go home, you search all the rooms, and you found the wallet. So that's the experimentation. The result is Wallace is found, okay? Or Wallace is not found. Okay? And then you compare that to the predictions of your hypothesis. Say Wallace found, the hypothesis is correct. Wallace not found, the hypothesis is wrong. Then you have to test a different hypothesis. So that is right there as the crux of the scientific method, okay? All right, now I'm going to give you an example of somewhat a realer scientific problem. Say you have some plants in your house, okay? And the mystery is you bought the two plants, at the same time, and yet the one that's near, that sits right here, somehow there it grows a lot better than the ones that you shoved like at the corner, away from the window. Okay, so that's the observation, right? And the question is, why is that the plant, that you place near the window grow faster than the plants that you took away at the corner somewhere? So that's the question, okay? So your hypothesis, right, you can generate different answers, right, or guesses. Say, maybe the plant right next to the window gets more sunlight, which then promote growth, okay? Or the plant on the floor away from the window, somehow you forgot to put fertilizers in there, so you just grow slower, okay? Or the plant next to the window gets fresh air, and therefore somehow fresh air promote faster growth, okay? And again, things like this, if you say there's an angel that lives near the window and that somehow talks to the plant and sing to it and make it grow faster, is this testable, that last one? Yeah, you cannot test it, so it's not scientific, right? So you have to come up with guesses or answers to the problem that are testable in order to be scientific. Okay, that's a critical, okay, component of doing science. Okay, so anyway, scientific hypotheses have to make testable predictions. So anyway, let's make a guess. Say it is sunlight, okay, that promotes growth. And that's why, that would explain why the plants next to the window growing better, faster than the ones away from the window. So what kind of experiments can you do? You go, the prediction, you have to create testable prediction. The testable prediction is that a plant receiving more light will grow bigger than the plant that gets less light, okay? And to do that, what you could do is you can just do a plant in full light, right, or next to the window, and then you could redo the experiments where partial light. You grow a pot of plants, not next to the window, but away from the window. And then for the control, you can just put the plant somewhere in the closet to make it dark, no light. So that's the experiment. So you perform the experiment, and you measure the height of the plant every day, right, or every 2 days, okay? And then you quantitatively plot it out, okay? Say, okay, number of days, okay, and then you plot out the height of the plant, okay? And say, right, that's what you do. You say light exposure is the number of days that's being exposed to light, and then the height of the plant, okay? And you get this. Say, the plants that have full light grows the fastest, right? Partial light grows the second fastest, and then the ones with no light fail to grow at all. And so that's your findings, right? And then you go back and you compare your findings to the hypothesis. And then therefore you arrive at the conclusion that the plant at the window grows faster because it receives more sunlight. So the hypothesis is supported, it's correct, right? But what if you get this result here instead of the previous results? Now you say, you perform the experiments and you get this. Actually, the ones with the partial lights grow faster than the full light. What's going on there, right? So that's the result. The plants in group B, the partial lights grow taller, more healthier, right, than group A, the full light. So your conclusion is, sunlight actually blunts plant growth. So my hypothesis is not correct, right? Okay, now, I'm just giving you the basic bare bone of the scientific methods, right, the different steps. When you work in the lab, real life problems, right, biological problems, the key step here, okay, is not just coming up with creative and relevant questions and coming up with hypotheses, right? That you can do, with training, with time. This step, experimental step, is very critical. Why is that? If you were to do the experiment like this and you get the result and you come to the conclusion that, sunlight is actually bad for plant growth, okay? But that here depends on how rigorous you perform the experiment. In the next slide, I'm going to show you that, okay? Show you how to do your experimentation carefully and rigorously. Because if you don't do that, you generate data or findings that are not strong enough, right? That allows you to make a statement, conclude whether or not your hypothesis is wrong or right. Okay. So, all right, the rigors of doing experiments. Okay. Sometimes you have to do the experiments multiple times. If the difference is, for example, light, let's suppose light enhances plant growth really dramatically, really significantly, then you might do a few experiments to come to that conclusion. But if light only promote growth marginally or like moderately, then you have to do many times to make sure that the conclusion is the data support your hypothesis. So it has to do with doing independent confirmations as well as sample size. Okay. Okay. When the effect is small, then you have to do a lot more. You have to increase your sample size. You have to do it multiple times to make sure. Okay. And there's also confounding variables. Okay. Sunlight is one variable that you're testing, but there are other variables that could impact plant growth, right? So it could be the fertilizers, right? If you do the experiment in such a way that you on the plant on the window, you give one type of fertilizer, and then the ones away from the window, another type of fertilizer, then you introduce another variable, right? You have to use the same fertilizer, okay? Plant type. In order to test this, you have to use the same plant, right? Same types of plant, okay? You can't use two different types of plant, because that can you introduce bias, and you introduce another variable. Maybe if you use, say, plant type X at the window, that tends to just grow a lot faster. That's much more higher growth rates than the ones away from the window. Then you can't really test your hypothesis on the effect of light, right? Water amount. You have to control for that too. You have to add whatever same amount of water to the two plants, okay? And then you have to account for days that you did the experiment. You want to make sure that the light is not rainy and cloudy the whole time, right? And then who knows, there are other unexpected phenomenon that you don't think about. Maybe the part of plant that you leave at the corner is more easily accessible to your pet dog, and he, for some reason, like to pee in it. And you don't know that. Maybe he does it at night. So you pee in it and say, that's nothing to do with your sunlight or water amount. It just happened that it just pee in it, and it kills the plant. Okay? So that's real scientific scenarios for you. When you work in the lab, the laboratory setting is where you're working on like cutting edge problems, okay? Problems that nobody in the world knows what that's why you're doing research. We do research because we don't know what the answers to that particular question is. So there's a lot of unknowns. So you have to, in order to do the experiments carefully, you have to control for a lot of things, okay? So that the effect that you see only depends on the variable that you test. In this case, is sunlight or light, okay? You have to keep all of these constant. Okay, so the concept of control, that's where we bring this to. How many of you, some of you have done research before. You know what controls are? What's? Yes?
Essentially like a baseline where you don't do anything with. So for example, in this case, I guess the control would be, I guess it depends on what your definition of control is. Like, a control could be away from the window where there's no sunlight at all, and have that base, and then add sunlight to see that changes. Or you could say, somewhere normal, like the living room, where there might be a little bit of sunlight, but not all, and then you see the differences.
Okay, yeah, that's a good start.
I was going to say, we have a negative and a positive control,
Mm-hmm.
and as we said, it's just a trend line or a baseline to see whether there has been a change in the experiment we've done. So we know the control, we know that this happened. While we do the experiment, we're looking if the subject is going to change, and we're using the control to then compare the result that we have obtained in the experiment.
Okay, all right, good try. So controls, yeah, you're on the right track. Controls, when we do controls, you have to ask yourself, controls for what? For what variable, okay? And controls, I'm sorry, what's your name again?
Julio.
Julio?
Yeah.
Okay, Julio just pointed out, controls are either steps or procedures that you know, right? You know the expected result. A positive control is when you know, oh, if I include this, I'm going to get an answer that support, you know, one of the question that I have at hand. Whereas the negative control, you say, okay, if I include a negative control, then I should not get that, you know, that answer, okay? And so that's what a positive or a negative controls are. And let me give you an example. Say, I want to control for the fertilizer component in the experiment, okay? So, for example, say Hunter, okay, perform the experiment, the plant experiment, okay? And when he applies the fertilizers, okay, he doesn't, you know, say he's very new at this. He doesn't know really how to, you know, how much to add, what kind of fertilizer to use, right? He might add, you know, a lot to one pot and little to another pot, okay? So he could add, you know, introduce a control for fertilizer. So he has, say, a different part of plant that he knows responds to fertilizer. Okay? So he say, okay, if I add fertilizer to this part of plant, right, and do it in a way that correct, then that's a positive control. Then that plant, right, should grow faster with the fertilizer. So that's the expected, that's a positive control. And then that same types of plant where you don't add fertilizer, it should not grow. So that's a negative control. So you have to control for what? You have to ask yourself, control for this, you know, for what? And in this case, it's control for fertilizing, the treatment procedure, okay? So you start out with that, right? And the positive control is if you apply the fertilizer correctly, you should end up like that, right? And then for the negative control for fertilizer, you don't apply, so that's what you should get. You expect to those results. And if you don't, for example, if you do this and you don't see this result, you see the same stunted growth, then you're not doing the experiments correctly. Because you expect the fertilizer to, right, to promote growth. So maybe I'm picking on you today, he bought fertilizer that are wrong. They're not fertilizer for a plant, maybe fertilizer for something else. Or he applied the fertilizer incorrectly, maybe too little or too much, right? So positive and negative controls, they're just controls for procedures and materials of a particular step in your experiment, okay? That allows you to assess whether or not you do the experiments correctly, okay? So that's what it is, okay? If your controls don't give you the expected result, then your materials that you chose or the way that you performed the experiment were wrong. So for example, how many of you know PCR? Okay. Or, you know, let me, the basic, okay. Or do cell culture. How many know how to do cell culture, right? Okay. So when you do the experiment, new experiment the first time, right, there are different components to an experiment. Say you do cell culture, right? And the control is, say, you want to control for a particular cell culture media, basically the food that you grow the cells in. So you want to test that, and you use some cells that respond to that media. If you use that media, it should promote cell divisions and growth. Okay? And you include that as a positive control for that particular media. Right? And if you do that and you see cell growth, you say, I performed the experiment correctly. But if you include that positive controls with that media and the cells didn't grow, there is something wrong with your procedure, your experiment, right? So it's very important to include positive and negative controls, especially when you perform a new procedure, a new experimental procedure. So let me give you an example, going back to the wallet thing, okay, the wallet example. Say if your hypothesis is I left my wallet at home, okay, and say I sent you, say, I'm sorry, what's your name, dear?
Alessandro.
Alessandro. Go home, go to my house and search the wallet, okay. So he goes, he searched for the wallet in the house, and he came back to me and said, oh, Dr. Vo, I searched, but there's no wallet. I couldn't find the wallet. So if I trust him, say, okay, fine. If you say you couldn't find it, then I must have lost the wallet in some other ways. But then, did he do the experiment correctly? Did he go to the right house, the right address, right? Did you search all the rooms, right?
Yeah.
And did you search, right? That's a big, say it's different rooms, and maybe I left my wallet in, I don't know, say in a trouser. Did you go through the trouser? Maybe you go through here, but the front pocket, but the back pocket is where the wallet is, right? So when you perform an experiment and a procedure, right, when there's multiple steps there, you have to do it right in order to generate data or findings that you can trust. Otherwise, if you don't do that, then the whole thing is shot. The whole research questions, the whole experiment, whole problem, you can't really make any conclusions about your hypothesis being wrong or right with shoddy data, okay? And especially in science, when you do experiments, it's important to include positive and negative controls when you're new at it, right? And you have to do the experiments multiple times in order to test your hypothesis. So very important. Okay, so that's why it's very hard, actually it's very hard to do science in general, okay? There are cases, there are problems that are very hard to confirm. So in fact, hypotheses can be proven wrong, but they can never be proven right or with absolute certainty, because it's impossible to test all the conditions, okay? So therefore, scientific knowledge, okay, it's very expensive. We spend a lot of time, a lot of headaches, a lot of effort and money to generate, kernels of knowledge, of information that are correct, right? Okay. But the information, we know it to a certain extent, but there's always some unknowns or some uncertainties there. Okay, so we could never be sure, and it's good. You guys know the famous scientist, physicist, Richard Feynman? Any of you? You okay. So he's the one who proposed this idea in that doubt and uncertainty is the core value of science. And if you think about it, it's true. Because when you always leave that, certain amount of uncertainty to your in your mind, then it allows you to probe some more and learn more about the problems rather than just take it as as truth. So that allows you to do more experiments to get closer to the truth. So anyway, what time how much time I have here? Okay, 15 minutes. Okay, all right. Anyway, the examples around you that you can ask yourself and do practice scientific. You can practice the scientific method. So, for example, if I were to ask Emma. By the way, Emma is Dr. Lee's daughter. I'm going to call you. Stand up, Emma. Kathy, that's my daughter. Stand up, Kathy. Okay, who's taller? Kathy. So that's an observation, and the question is, okay, why is Kathy taller than Emma? So that's the question, right? And what hypotheses would you come up? So give me give me a guess.
Me? She drank more milk as a kid.
Right, that's a good hypothesis.
I was saying she's older.
That age, yeah, that's another hypothesis, right? Okay, or just Kathy's dad abused, her a lot, so she overeat as the results of the constant abuse, and therefore she grows bigger. So there's a lot of ways to come up with your hypothesis, right? And then, but they have to make testable predictions. Okay. So if, for example, age, you can test that. You can go to the birth record and compare, right? And then milk, you have to go back and see, the amount of milk consumption from the two of them. So then you see, you see how hard it is. So it's the biological problems that you work in the lab. They're not simple, straightforward. They can be very tricky, and you have to do the experiments correctly, okay, in order to test the hypothesis. And even then, you still need to do some more. Okay. So anyway, here, and remember, use this framework, and that will help you a lot. Okay, to do all of this. When you read papers, when you listen to a research talk, when you write, papers, and when you present, this is a very helpful framework. That here, the scientific method. Okay, to allow you to assess how science is done. Okay, so for instance, say you read papers, say, what is the question, or what's the problem being studied here? Then what kind of what hypothesis do they? Sometimes papers, they have implicated or they imply some hypothesis, but they're not written out. So you have to you yourself, as you read papers, you have to come up with that, right? And again, it's the quality of the paper too. If it's a good quality research paper, you read and say, it's very clear. This is the scientific problem. Okay, and these are the hypotheses and with this specific testable predictions. In fact, the figures that they embed in the papers, those are designed, or those are findings that test the predictions of the paper, of the study. Okay, and then you read and you say, okay, these are the findings. Okay, do they really match? Do they really, agree with the testable predictions, right? And you have that in mind in order to critically evaluate the study. Same as you sit through a scientific talk, you listen, right? And you go through these kind of steps here to see if whatever the lecturer or the speaker present, is that, good science is the data done, generate from the right types of, rigorous experiments, right? So you could evaluate. So I'm giving you kids a tool to evaluate science, okay? All right. Yeah, anyway, if you stick with biology, I'm biased. Biological structures are just marvelous, like some of the most mysterious things that you're going to learn. Okay, there you go. I just gave you the golden key to living a happy life. So keeping a lab notebook is very important, okay? And the reason for that is this. Say, let me see. Let me ask you, what did you have for dinner last night?
Last night,
Yeah.
I had spaghetti.
Good. What did you have dinner for, like, last week? A week ago?
That's a great question. I don't know.
See? So that is the reason why you keep a good lab notebook. It's because of that. We could only store so much here, right? And then, a few days later, if you don't write things down, you're gonna forget. There's no way you can retain all that information. So keeping a lab notebook is very important because it provides proof of record, right? And you can trace and repeat the experiment, okay? For instance, when I was a graduate student, I went and talked to my PI and say, oh, Dr. Chamberlain, I want to do this experiment. How do you? And he say, go to so-and-so like John's book. We used to keep the lab notebook, like physical lab notebook. Go to, that book number 10, go to his experiments 55. And lo and behold, actually, there's like half a room full of notebooks, and somehow the guy remember all the notebooks and where. And I went there and I opened, lo and behold, there it is. That's the procedure that I want to do with all the details. So it's very powerful, okay? To locate reagents, recipes, protocols, etc. And the information are in your lab notebook. And it's very important for you. And it's very important for the PIs as well as your peers, the other lab members, okay, to follow, to troubleshoot with you, for instance, right? Or to, you can share some successful experiments with your peer later on, right? So, all right. So for a research project, you keep lab notebook, one or more lab notebooks, okay? And these are the basic components of keeping a proper lab notebook. And I didn't come up with this. This came up by some Nobel Prize guy at Stanford who passed on to my PI, and my PI passed it on to me, and I passed it on to my students. And these are well tested, like time tested. So it's very, it worked, okay? So what you have is normally, your name, the date that you perform the experiment, okay? And then typically you write down, it's like the experiment number is like a code. So that way you can refer to experiment like 61, for instance. You can reference it, okay? And then the next two components, the goal. You have to put down what experiment you're doing. You have to describe what it is. Then the next one is the purpose. Why are you doing it? If you just write down what the experiment you do without giving the rationale, a week from now or a year from now, say, I'm not sure I'm not sure why I did this experiment, right? So you write that down. And then for new experiments that are new to you, normally I ask my students to go through what's called a preparation step. That's where you try to do some research and look up recipes, references, make flow charts. For example, if you were to, I don't know, we don't do this anymore, but purify a protein or learn how to do PCR for a newcomer, right? It's actually pretty intimidating. There's a bunch of steps. So this allows you to make outline and understand conceptually how the flow of experiments, the different steps. Okay, and then the procedure. You have to write down step 1, step 2, step 3, step 4. Write those down, okay? And then, you know, if you repeat some procedure that you've done before, instead of writing all those down, you can copy and paste if it's electronic notebook, or you can reference, say, I performed using the procedure from experiment 31 in that notebook. So you can do that, okay? And the result, here, this is what you have to put down, what the result, right, what you get. And in general, there are different types of results. If it's imaging result, then you have to annotate. You have to label the lanes, or you have to tell what color, what they mean. You don't just plop the thing in their notebook and do not explain, right? Then you go back again. Like you could remember what you did for about a week or maybe a month, but half a year from now or a year from now, you're not going to remember. And you look at that image and say, what is red, or yellow, whatever the images, the structure mean. You have to annotate. And then the conclusion could be writing key findings and making some conclusions. And if you make mistakes or see weird result, mention that, so that when you go back, say, I think this weird result, I might have gotten this from this step, where instead of adding 2 microliters of enzyme, I mistakenly added 5 microliters, or something like that. Okay, so that reminds you. Okay, so this is actually I'm giving you an example here. Say I did the experiment on that day, right? And this is experiment 1. So my goal, the goal of experiment, is to purify total RNAs from the old disk. Okay. And if I don't put down the purpose, meaning the rationale, I wouldn't, a month or 2 from now, I wouldn't be able to remember why I purify the RNAs for. You have to put down, I did that because I want to do gene expression profiling, right? I use these RNAs in a technique called RNA microarray experiment to look at all the expression of different genes, okay, in this tissue. And that's the reason for doing so. And then you go through some preparation. And since I was new at this, I had to list down the flow chart experiments. And these are really good exercise. Then you have the concept, the conceptual design of the experiment. Okay, especially when you're new with the procedure. It helps a lot. And then, of course, then you put down the procedure, right? And then the results, you plot out either in table form or in graphical forms or images, but you have to label and annotate, okay? And then you do that, and then conclusion, you say, yeah, I got about, roughly the yield is 3 micrograms of RNA per 10 milligrams of tissues, and it seems like young tissues tend to give higher RNA yield. And then I say, there's some weird result, and I can put that down. But if you don't have weird result, you don't have to put that down, okay? And the conclusion could be, very short. It's only long when you have some strange result. And then if you don't have any mistakes, you don't have to put that down, right? And mistakes and improvements, you say, be careful next time when you do step number 2. It's the extraction, is the removing the fluid from the pellet. If you don't do it right, you can contaminate your sample, for instance. So things like that. But if you don't have that, then you don't have to put it down. But putting key, information down in your lab notebook. And if you learn, you discipline yourself and do this kind of recording, is extremely important. Okay, that's a what is it? It's a trait of a good scientist. Okay. Okay. So, any questions? So what I want to emphasize here is doing science is fun, okay? But doing science is hard. It's very hard to do good science. And you have to acquire all these skills, right, in order to do rigorous and do good science. Because Mother Nature, she's pretty secretive. It's very hard to extract information from her. So learn these, technique, and I hope that you go away with this scientific method in your mind. Okay, so that'll help you evaluate, research in general. When you read a paper, a newspaper, for instance, or just social media stuff, you have to ask yourself those, questions. You go through the scientific method and see, evaluate what you read. Otherwise, you're just gonna, absorb these, social media information that a lot of them are wrong. So this allows you the ability to evaluate, information that's just like flowing all over out there, especially now in this days and age. And then the second thing is the keeping a proper lab notebook. Okay. All right. Questions before we end?
I guess I would add one thing. So the discussion of controls, I think you talked a lot about experiment. You applied many kinds of controls. One we focused a lot on was what I would call the experimental control, to answer the experimental question.
Yeah.
And one thing I find many young people don't want to do, or in the procedures, we outline and understand the concepts of the experiment. Many of those steps have internal controls to tell you that you did the experiment properly.
Yeah.
And I call those technical controls. I'm not sure that's the real word for it, but
What's a?
I call it technical, like making
technical
sure that you
controls,
do the
yeah.
experiment properly. And they take time.
Yeah.
They use reagents. They can be highly frustrating, but they're super important. And many people make many shortcuts in that. That would encourage. Yeah, you spend more time in that experiment the first time, but you'll know you have a result as opposed to wondering whether you have a result. So that.
Yeah, if you do, say, let's take an example, like you do an experiment is only one step, right? And if that step, is simple, then yeah, you just do it and it's not too bad, right? It's straightforward. But if the experiment involves 100 steps, okay, and step 10 depends on step 1, or step 30 depends on step 21, right? It's experiment like that where you do, and if you don't say you pipette, you're supposed to pipette 2.5 microliters of something into there, and at that step you pipette, and instead of 2.5 microliters, you pipette like 3 or 4 microliters, right? Then you didn't really do the experiment properly. And then, therefore, the result you get at the end, it's affected by that particular step. Okay? So I used to do what's called protein purification in the past. And it's like, 100 steps. And so I spent 2 or 3 months purifying this tricky protein. And you have to be very careful at different steps, because if you messed up any of those prior steps, at the very end, you look at the result, and back then, you have to, what's called fractionation. You have to put it in, your protein, purified protein through a column, and you collect these little tubes, little amount of fluid as the sample goes through this column. And then you have to put those little tubes into a reader. And if you get like a peak at the right position, that's purified proteins. And after 3 months of hard work, and when you get there and you don't see the peak, then you're very sad. You become depressed for about a week because you spend 3 months of hard work and you don't see this because there's certain steps in that in the procedure that you didn't perform. I didn't perform correctly. Okay.
Is there no way to check between each step as you're doing it in those 3 months? So then this way, like next time you know, rather than like, oh crap, I wasted 3 months, and then we just redo it and like try to be more careful.
Yeah. So, yes, there. When a procedure, when you deal with a complex procedure, you have to do it with somebody, right, initially, to guide you through, like somebody who had gone through that before, and point out to you those steps, especially the tricky steps, that you have to practice with that person to do it correctly, right? So that's one way to do it. Normally, when you start out with some simple procedures, and even that, still, the way science is taught is, okay. Somebody, for example, Pete, Dr. Alexander, show you how to do it, a new procedure, okay. He's gonna do it. You watch, okay, and then you do it together, and then you do it. He's gonna watch you, right? And then after he evaluated that you do the procedure correctly, then you could do it on your own. So that's how it works typically in a lab. You have to go through that steps. You watch, okay, then you do, and you being watched, and then you can do the steps on your own. All right, good luck. Have fun.
Thank you, Dr. Bao.
You're welcome.